Welcome back to Biology Boost Up, and in this video tutorial session we going to study about the types of DNA, the repetitive DNA, and a special topic at the end of this session. So now let's start. At first, on the basis of shape, DNA may be also two types, the circular DNA or the linear DNA, depending upon the presence or absence of free end. In case of circular DNA with no free ends, closed covalently at its two ends, occurs in nucleate of bacteria and randomly in nucleate of semi-autonomous cell organelles like mitochondria, chloroplast, etc. Whereas in case of linear DNA has free ends, occurs in eukaryotic nucleus, some prokaryotes like PPLO, and some autonomous cell organelles. Next, on the basis of number of strands, the DNA may be classified into single-strand DNA and double-stranded DNA. In case of single-stranded DNA, it was first discovered in the spherical bacteriophage. In case of double-stranded DNA, it contains or consists of two antiparallel strands of deoxyribonucleic unit. It is found in almost all cells which DNA act as a genetic materials. Now the next topic is repetitive DNA. It is the part of DNA containing the same sequences of nitrogen base repeated several times in tandemly. It occurs near telomere, centromere, pericentric and heterochromatic area. It is postulated that it functions in chromosome pairing during meiosis, crossing over, protecting of important structural genes. The satellite DNA is the part of DNA which has long repetitive nucleotide sequence as tandem areas that forms a separate fraction on density ultracentrifugation. The satellite DNA can be two types. The microsatellite sequences, that is 1 to 6 base pair repeat units flanked by conserved sequence, and the second type is mini satellite sequence, that is 11 to 30 base pair flanked by conserved restriction site. It is very useful for genetic mapping because of high variability. Now next, the functions or role of DNA inside the cell. At first, DNA is a genetic material which carry all the hereditary informations coded in the arrangement of its nitrogen bases, that is genetic code. The second is DNA gives rise to RNAs through transcription and control the metabolic reactions through directing RNAs to synthesis of different types of proteins like enzymes. The third point is DNA functions two important roles. First, the heterocatalytic functions and the autocatalytic functions. In heterocatalytic functions, DNA directs the synthesis of chemical molecule other than itself, that is synthesis of RNA or protein. In autocatalytic functions, DNA directs the synthesis of DNA itself. Now, the special topic is why a base pair must contain a purine and pyrimidine nitrogen base. There are two reasons for this limitation. The space available between the two sugar backbone chain of DNA, that is 20 Armstrong, can be accommodate only one purine and one pyrimidine, but not two purines, which would be too large, and not two pyrimidines, which would not be closer enough to form proper hydrogen bond. On the other hand, a specific purine pairs with a specific pyrimidine because of a perfect match between hydrogen donor and acceptor site on the two bases. Only adenine and thymine and also guanine and cytosine have the proper special arrangement to form correct hydrogen bonds. Now next, the types of DNA on the basis of structure, or you can say the difference between bDNA, zDNA and aDNA. Now look, the bDNA is right-handed helix. In case of zDNA, it is left-handed helix, and aDNA also left-handed helix. In case of bDNA, the pitch of helix per turn is 34 Armstrong, whereas in case of zDNA, this is 45 Armstrong, and in case of aDNA, this is 25 Armstrong. In the third point, the diameter of helix in case of bDNA, it is 20 Armstrong. In case of zDNA, it is 18 Armstrong, and aDNA, it is 23 Armstrong. The fourth point is the stability. The bDNA is stable and physiologically active, but both the zDNA and aDNA are unstable. Now next, the base pair per turn of helix. In case of bDNA, it is 10. In case of zDNA, it is 12, and in case of aDNA, it is 11. Sixth, the distance between two base pair. In case of bDNA, it is 3.4 Armstrong. In zDNA, it is 3.8 Armstrong, and in aDNA, it is 2.5 Armstrong. Now the last point is repeating unit. The bDNA is mononucleotide, the zDNA is dinucleotide, and the aDNA is also mononucleotide. Thanks for watching the video. Please like, share and subscribe the channel.